Okay, I think we're ready to get started for the continuation of the HRC reports. In this session of genetics and breeding, we will have John Henning, and John is with the USDA-ARS located in Beaver Country. It's also known as Corvallis, Oregon. So welcome, John, and we'll allow you to give a little recap.
Okay, thanks, Brian. Get my screen up and ready. And in order to do that, let's okay. Thank you all for being here. I noticed there's a large number of people today, and that's great to see the interest in what's going on in the research field. And just going to go over a short summary of some of the projects that we've been doing, and you can see more in the actual recorded message. So this past year, we made a series of 20 crosses, and most of these were based upon basically aroma and alpha. This is a list here, and you can see this in the show, but we're not going to talk about it. But basically looking at increasing powdery mildew durability, looking at replacing some of the, I call the 3 Cs, the Cascade, Centennial, and Chinook. In addition, trying to make some crosses that might expand where we can grow hops below the 35th parallel with the Southern Brewer. And so these are the number of the seeds that we came out of that, and we're going to be germinating these this year and hopefully getting them into the cycle soon. As a reminder, the 2018 nursery was a set of crosses that I made to try and work on development of downy mildew in population basis rather than simply pedigree breeding. And so what this consisted of was selecting a series of females that have good tolerance to downy mildew and crossing them with what is basically a bulk of pollen from downy mildew resistant or tolerant male lines. We started off with quite a few seedlings at that point. I think it was about 12,000 seedlings, and now we've whittled that down to 93 individuals, or we're going to try and get it even down further than that. Where we'll have 2 representatives, hopefully a male and female from each one of these families you see listed on the bottom here. Moving on to the 2017-2016 nursery, we now have 351 offspring that were selected, and they were transplanted this past year, 2020, out at the 4B farms on single hill trials. They were staked up with bamboo. These have been scored for powdery mildew and downy mildew, and we didn't see very much of those. So the hope is that we did a good job in the greenhouse and screening those susceptible ones out. They're going to be strung for harvest this upcoming year for the first year in 2021. Moving on to the 2015 nursery, which is again single hill nursery. As a reminder, this was a series of crosses that was made several years ago, and just due to circumstances and various sort of efforts that were going on at the time, we did several rounds of selection for downy mildew resistance in this group. And the offspring that came out of that is 26 selections that were planted out in the field. And each one of these selections actually is a very, very strong plant. They're pretty vigorous plants. Unfortunately, most of these seem to have a harvest period of about mid-September. And those of you who are not aware of this, Oregon and Washington had some extremely severe fire season this past summer, and it actually happened during harvest time. And these plots were actually in restricted zone that could not be driven to because they were so close to the fires, and the smoke and the air quality were so poor. We did manage to get 4 of these harvested, and this is sort of a summary of what we saw. Moving on to the advanced nurseries, these are nurseries that have up to 15, 16, and up to 20 hills per line. And so we have 32 of these lines at Fobert Farms near Hubbard. And we have several, also 9 present at 4B Farms. And not all of these were actually, again, able to be harvested due to the significant smoke damage that came about, because these lines were, the ones that didn't get harvested were later, and they were exposed to smoke, and just you could really smell it on the hop cones when we were smelling it. So these are what we saw. The ones that are in red actually yielded quite well, look promising, but we'll give them another year to see how well they can do. The selections from the 2014 crosses are in 5-gallon pots, and they were planted in 2020 up in Washington, but they'll be planted this spring in Oregon. Those of you who were not able to actually listen in on Kayla's and my talk on Monday, I'm just going to briefly talk about 2 of the elite lines, 2 promising lines that are coming up. So this is the part where I shamelessly promote my own varieties. So elite line grow out. The first one is 2010-008. This is being grown currently at Crosby Farms and Puderbar Farms in Washington. It's an extremely early, high-yielding line. And the benefit of this is it really aids the farmers by not having yet another variety smack dab right in the middle of harvest, but it allows them to begin their picking season earlier. It has a floral, fruity, noble hop style. Currently, there's about 3,000 pounds of pelleted available through Brewer Supply Group, Hollandsberry, Mill 95, and Crosby hop. So this is more of a Pilsner and lager style hop that you might use, or just general all-purposing bittering hop. It's not high alpha. The next one is 2006-009-074. This is actually a triploid line, so it's a seedless line. It's currently being grown at Siegel Ranch in Washington and Capitol Farms in Oregon. The theme about this one, it's extremely high yielding, very, very vigorous seedless aroma line, and all of the brewing reports that have come out over the years have been extremely favorable for a unique IPA style hop. It does have broad adaptability across the United States. It's actually being grown all the way from the West Coast all the way to the East Coast, from Washington to Vermont, and all the way down into California. It does seem to have some drought tolerance and heat tolerance, is what we saw in 2015. There's approximately 3,000 pounds available of this one too. And of course, for both of these elite lines, we should see more of them next year, because this was basically the first year of harvest for most of them. So moving on into some of the genomic and genetic studies that my team and I were able to accomplish. I'm not going to spend a lot of time, but Dr. Nala Basil and myself have finished DNA fingerprinting studies and are in the process of writing up a paper on that offers 2 different methodologies to do DNA fingerprinting of the hop varieties that are present. We also are working on and near completion of sequencing and fully assembling 2 new genomes of both of them are male hop lines. We completed the assembly of the 10 hop chromosomes. So basically now we have all 10 chromosomes assembled into units, and we have identified all the genes and mapped their locations on those chromosomes. Working with Dr. Jave Gent's group and Michelle and the postdoc, Renee Erickson, we are working on an RNA-seq of powdery mildew resistance over time, and just looking at what genes are operating under those systems. And the last 2, I'm going to just spend a brief amount of time talking about, but that would be the physical and genetic mapping of quantitative resistance to powdery mildew and downy mildew, and then also looking at the genomics of abiotic stress tolerance. So Michelle Weissman spent a lot of time phenotyping a large population of hops, about 300 individuals, in the greenhouse for multiple times. And we had excellent phenotypic data. And we also did the genomics work on all of these individuals of a cross by Comet by 64035M, Comet having the quantitative resistance, or what we suspect is quantitative resistance to powdery mildew, and 64035 having the quantitative resistance to downy mildew. The interesting thing that we found about this is typically when you're talking about quantitative traits, you're seeing the genes that are involved in those traits spread across the whole genome. We did not see this for powdery mildew quantitative resistance. In this case, we actually see one extremely expressed region right there on chromosome number 7. It covers 5 megabases or 5 million bases. And within those 5 million bases, there are 36 R genes and 26 disease-related genes. So somewhat of a new idea of how powdery mildew resistance in Comet is working and functioning. The other work that Renee Erickson has been working on is just looking at what are the effects on drought on the alpha acids, and in particular, we also looked at other chemical compounds, but I'm just going to report on what happens to alpha acid. So what we see under high temperatures or low water conditions, or the combination of the two, is that there's a severe and significant decline in the filarophenome synthase and the humulone synthase expression. And these are the genes that are responsible for alpha acid production under stress conditions. So when you have low water conditions or high heat, or even the combination of the two, the expression of those genes making the alpha acid is significantly reduced. So simultaneously to that, we also looked at when these genes are most important in the development of the hop cone. And what we saw was that the mid-season development is the most critical period for the final stages of bitter acid production. So these genes are at their highest expression during the mid-stage of cone development. And I think the take-home message for this is that there's no way that we can avoid the heat during this time, but I think the important thing to do is make sure you have your water during the mid-cone season development. So none of this could be possible without all of the different people that are involved in here, and there's way too many people to sit there and talk about, but all of them are definitely important to what we did. And with that, I just want to thank you, the Hop Research Council and the State Hop Commissions and the Brewers Association, and also hop growers, for all of the great work and encouragement and support that you've provided.
Okay, thank you, John. So if you have questions for John, go ahead and type those out in the Q&A box. And then what we'll do is, after Kayla's presentation, we'll try to answer a couple of those questions quickly, and they'll do that jointly. So next presenter, Kayla Altendorf, who is the newest member to the HOP group, and she started with the USDA-ARS. She is located in Prosser, Washington, and she started this year. She's been on a very, very steep learning curve and has had several very unusual events disrupt that a little bit, but she is learning rapidly. So Kayla?
Thanks, Brian. All right. So thanks so much for the opportunity to present. I didn't have a project that was specifically funded by HRC this year because it was my first year, but I really wanted to take the opportunity to write up a report and document the initiation of this new breeding program. Looking back at previous HRC reports for the Washington State breeding program has been really an important part of me getting established here in Prosser, and I know the importance of those records. So I wanted to contribute and document this new program. So for those who I haven't had a chance to meet, my name is Kayla. I'm from Minnesota, and I got my master's and PhD degrees from the University of Minnesota. And a lot of my work in graduate school was focused on the domestication of a perennial grain crop called intermediate wheatgrass. So a lot of experience working in newly domesticated species, but also was a member of the wheat lab at the University of Minnesota. So like Brian said, I was new to hops in May, moved into to Prosser in May. And many of you know about the breeding program in Corvallis, obviously, but in 2018, Washington State University signed over a material transfer agreement with the USDA to allow the USDA to utilize the existing germplasm as part of the start of this new program. So the program is located right in Prosser, just north of town, at the Irrigated Agriculture Research and Extension Center, which is a part of Washington State University. And at the time of the initiation of the program, there's a lot of germplasm already at the station. So 158 lines that were developed by Washington State by the former breeder, Steve Kenny. 107 lines that were developed at the USDA in Corvallis by John. And these lines that are being evaluated as advanced lines range from cross years from 2000 to 2015. So it's quite a backlog of germplasm being evaluated currently at the station. And these plants range, or these plot sizes range in size between 3 and 59 hills. So again, quite a bit of variation in terms of size, and also spread across 4 different yards, both at the main headquarters station as well as north of the station at the Rosa Farm. And then there's also a wealth of parental germplasm. We have over 200 males and over 400 different females that are intended to be used for crossing. And over the next few weeks, I'll be collecting some more lines down from John's program just to get a complete set of germplasm that I can use for crossing here. So in my HRC report, I just kind of highlight what we've been up to for the first few months. And I'm just going to really briefly recap some of those activities. So we've been working on making connections by visiting farms and breweries and propagators. And I just want to thank everyone so much for being so welcoming to me. It's been great to meet you, and it's been kind of a difficult time, obviously, to get established and make connections in the industry, but people have been really tremendously welcoming to me. So I'm grateful for that. Also working on establishing the lab. So I hired a technician, Anna Tauril. She's from Michigan, and she's been a great addition to the team so far. I'm super grateful to have her. And then I also have a postdoc opening. So if anyone's interested, I really encourage you to reach out to me or pass along the information. I'm going to be evaluating those starting this week, or evaluating the applications. So I really encourage you, if you're interested, to apply really soon. And please spread the word about that. Also working on undergoing some renovations at the station, working on establishing the male yard so that it can be replicated and randomized and can be a source of different studies to look at the males. Like I said, we have over 200. I'm looking to expand that with some of John's males. And I'm excited to have this yard to begin evaluating the males, and it can provide an opportunity for lots of different studies. So I invite collaboration on that if anyone's interested. Also been working on increasing our technology, or the use of technology in the breeding program by implementing tablet-based data collection, barcoding, and my technician Anna has been working hard to develop us a database that we can use to house our data and establish a long-term data storage plan. I also, like John, made 20 crosses this year. I was new to crossing in hops, so it was an interesting learning experience, and I'm really grateful to all of those who've helped me learn, mostly over text message or email. Many of them were successful, so I was super grateful. I did lose a few to the wind on Labor Day, but I'm excited to get these planted out and begin evaluating for disease resistance and making selections. Another thing John mentioned in his talk, that we established a trial here in Prosser of some of his advanced material. And new this year is I implemented some experimental design attributes into the evaluation procedure, which I'm super excited about. So we started with rhizome cuttings, and in the middle of the figure, you can see where, for example, each line would be represented by a different color. And historically, we would receive a variable number of rhizomes per genotype. And so in some cases, some plants would be represented by a really large plot and some by a really small plot. And that just makes it really difficult in terms of record keeping and tracking, because there's variable plot sizes that makes it complicated to calculate yield. So this past summer, we established the first advanced trial in this manner, in the randomized, replicated manner with border plants as well as commercial checks, so that we can account for seasonal variation. And I'm really excited about this moving forward to be able to conduct statistical analyses, for example, on the data that comes out of the breeding program, which I think will allow us to make more informed decisions more efficiently and with greater confidence in terms of advancing and selecting and culling different lines. Also working on establishing some more off-station trials. So we've got one in the works in Idaho with Justin Clements, and been getting a few emails over the last few days about other people who are interested across the country. So looking forward to getting our germplasm evaluated in more environments. Another thing I've been working on is taking inventory and starting to advance some of the existing material. I mentioned that we've got quite a backlog of some really promising material at the station. So on the left-hand side, I'm showing the hop source data. So I inherited basically 2 sets of data for the program, just ranging from like 2017 to 2019, both looking at yield as well as the hop source data. And in this figure, I'm showing just the hop source data, and I'm showing them by rank. So on the top, we have the highest ranking individuals, and the bottom would be the lowest ranking individuals. And lines that were evaluated in more than 1 year are connected by a same color line. And normally this plot is like an interactive plot where you can hover your mouse over a different color line and the name of the genotype will pop up, but didn't quite figure out how to add that into, I'm sure there's a way, but into PowerPoint for this talk. But if you're interested, I'm happy to share this figure. But the takeaway message is just that there's been a lot of investment in evaluating some of the material that exists here in Prosser. And as you can see, many of them were evaluated in more than 1 year. So theoretically, we should be prepared to make some decisions based on this data. And you can see there's a lot of crossover. Again, not surprising considering it's a very subjective trait in terms of like aroma and preference. But just looking at trying to identify the individuals that ranked really well in multiple years. And I'm doing the same thing with the yield data. There's some limitation just with regards to the replication and the number of lines that were evaluated, and some weren't evaluated every year. So it's a little tricky to make decisions based on this, but so that's why I'm looking at individuals that show up repeatedly as promising, and then working to move those into some more advanced and replicated trials, because a lot of these lines have been stuck in like the 3-hill plot format for a while, and looking forward to getting them bulked up and evaluated more closely. And then of course, initiating some new projects and collaboration. So it's been mentioned a few times, but working on evaluating the male yard for mite resistance with Doug. Working on kind of taking inventory of the public hop breeding programs with collaborator Josh Havel at the University of Minnesota. And we're working on an app that can be used to calculate co-ancestry to try to make parental selection easier and more efficient. And then working with Dave and John and other collaborators on establishing some studies for QTL mapping for disease resistance. So like John, I have also a lot of people to thank. My collaborators within the ARS and the University of Minnesota, as well as the Walsh Lab. They've been a huge help for me in getting established here and helping to maintain the wealth of germplasm that we have. And then obviously thanks so much to the HRC for providing lots of guidance as I've gotten started and providing the historical data and providing me the opportunity to present today. So I also just want to thank the Brewers Association, and I also am really grateful we have a public hop breeding advisory committee, which I think is really great, and I enjoy communicating with them. And then again, just thanks so much to everyone who's invited me for tours and allowed me the opportunity to increase my knowledge of hops. So thanks so much, and I look forward to answering questions.
All right, so if you have a question, go ahead and drop it in the Q&A, and I can read those off to John or Kayla.
John, I'll start by just asking a quick question about the Elite Line program. So we are trying to increase the number of selections that are in the Elite Line program with the intention of just having more selections out there at one time for evaluation. It's meaningful information that gets fed back to you that you can help in that process of determining whether to release a selection or not. In your thought process, how many years do you think these selections are going to be in the Elite Line program?
Well, I think that, the first year is sort of a wash, but the second and third year are definitely critical. The hope is by the third year that we have a good understanding that brewers like something and that they want it to be expanded even further. So the goal would be by the third year to have it to a point where we could have it released. That would be the goal.
Okay. And we have two of those selections that will be going into their second growing year, essentially production year, this next year. And both of them have received very, very good comments.
Yeah, it's exciting. I mean, can't wait to get them out there into a lot more brewers' hands.
We've got a question from Ryan. Do you have an idea of the number of reps and randomization designs that are best for hop yield trials?
Kayla, you want to take that?
Yeah, sure. I went with 2 this year just because I mentioned I have a lot of germplasm and I really have just a small field space left to expand at this time. So I chose 2. I definitely wanted more than 1. Didn't really have space for any more, but I don't really have a ton of insight on how many one should have. I think from a logistical standpoint, in terms of like harvesting them in multiple reps, the more you have might get even more complicated. I know in my case, just with how many we're harvesting every year, my staff was a little reluctant to add replication. So yeah, I think that's why I landed on 2, but I don't know. John, have you ever done more than 2 and found it valuable?
No, I haven't, but I think you hit it right on the button there. It's the more replications you have, the basic more locations that the workers have to go to get that line, just adds to the crush of harvest, which is full on. So.
Yeah, I mean, if I was just doing like looking at one tray, like in a mapping population or something, maybe I would consider more. But in terms of advanced field trials where you've got multiple lines, I'm going to stick with 2 at least for now.
Yeah, I would recommend that myself.
And as far as randomization goes, I mean, you got to do it.
Okay.
Right on time, without any other questions, but if more come in, I'll alert you to them to answer in the Q&A box. But other than that, I think we're caught up on questions.
Thank you all.
Thank you, John. Thank you, Kayla. Appreciate the summaries.
Thank you.